Good morning. I'm Rajesh Ambasudhan. I'm Chief Strategist of Allele Biotechnology in San Diego. I've been attending these events for the past several years. I'm so glad that few people remain in this room. I won't disappoint you. The program is coming to a very close end. ARM possibly has kept the best for the last, just like a showstopper in a fashion show. So I'm going to try my best to live up to that words and start with my talk here. Oh, what happened? How do you move forward? Okay. So at Allele, we do things, we try to stay ahead by doing two things. One is to try to recruit people who are immensely talented and who has a wonderful background. Second, we try to invest on programs which clearly distinguish us from the rest of the crowd. So everything what we do in Allele is using mRNA technology, except for the first two items which described on the top of the slide, nanoantibodies and fluorescent proteins. So nanoantibodies, as you know, they are very tiny single-domain antibodies naturally occurring in camelids. And we have a llama farm which produces this. And we have a wonderful colleague of mine who is being trained, not with one Nobel laureate, but two Nobel laureates, Richard Axel and Susumu Onagawa at MIT, who is leading this program. We have a lead program on this, which is targeting TNF-alpha and IL-23 bispecific for inflammatory bowel disease. The other program on the right, extreme right, fluorescent proteins, again, we are not a GFP or YFP producers. We are a unique fluorescent protein producers and fluorescent reporters. This program is, again, led by another colleague of mine who is trained with Roger Chen, who invented the GFP. So he has a unique, very rare kind of a hobby, going to Australian coral reefs, deep dive in the ocean and go to the ocean bed and try to collect all those curious creatures and organisms from which we can isolate the fluorescent proteins. And we are very proud to say that in the recent COVID pandemic, our fluorescent proteins were used as a means to discover the therapy by Pfizer, Moderna, as well as Regeneron for their viral vaccine programs. So apart from that, we do everything using mRNA. And our main mRNA focus is on iPSCs, and we try to generate these iPSCs in the GMP manner. We have several off-the-shelf lines of iPSCs. And what distinguishes us from the rest of the crowd who make iPSCs is that our iPSCs is IP-supported. It's the mRNA-driven, fully mRNA-driven. And we produce it in our GMP suite, which is custom or niche built for iPSC manufacturing. We spent a lot of years on building this facility. We bought a facility in San Diego and tailored it for GMP, iPSC-based therapeutic development. We do strict donor eligibility testing of the donors who come to us. We have a clinical suite inside where clinicians sit and do the biopsy. From that point onwards, we have full chain of custody of the item, and we and subsequently to make fibroblast and over to iPSCs. Everything is under an unbroken chain of GMP. Third, we have an industry-efficient method of producing iPSCs with stringent quality management system built over several years of hard work for some of our colleagues. So we believe our iPSCs are extremely robust and competitive as opposed to many others making with the episomal and other competing methods. So our iPSCs, as I mentioned, it's a very robust system. We make, as opposed to 3 to 4 weeks of generating the first colony when you reprogram, our cells emerge in about 8 to 9 days. And then they are without genome integration, and they are viral-free, they are CNO-free, they are consistently, you can produce this. And after expanding them over a brief period, a couple of passages. They're ready to be used for your purpose. And some of our lines have been luckily licensed by companies like Astellas, and ironically, many of our customers are from Japan, which is a hotbed of iPSCs. So I won't go into those details. The way we do is mRNA, as I told you. We sequentially introduce mRNA into the cells. Our speaker from GC Bio earlier mentioned about the power of transcription factors. So in this case, we use the mRNA for transcription factors, and over a period of several days, keep introducing them to make iPSCs. This is not trivial because many people tried, and it is the cells may produce an interferon response and other complications. But we have mastered a way by which we can produce, and we can be much more efficient and safe as opposed to conventional method of DNA using DNA vectors. So not only we make iPSCs, we also differentiate the cells using mRNA. So we, for mRNA-driven reprogramming to make iPSCs, and then we use those cells with specific transcription factors encoded by mRNA to produce lineage-specific cells. The ones which I highlighted here, pancreatic beta cells, hepatocytes, and all those in red, are the programs we are currently using them in therapeutic purposes. So before going into, I will focus much of my talk on the beta cell program, but before that, I will show you, spend a couple minutes on this slide, which is most exciting in our company right now. Yesterday, Aspect Bio was discussing about making tissues from iPSC or stem cell-derived tissues bioprinted for human therapeutic purpose. We are not making tissues, we are making organs. And what I meant by organs is that from iPSCs, we differentiate all different cell types of the liver, the hepatocytes, Kupffer cells, stellate cells, and endothelial cells in a specific ratio and manner. And then we print them along with blood vessels and biliary ducts into a scaffold, and we introduce into animal for the transplantation. So even this is an early program. We are lucky that government is with us. We have been funded by ARPA-H. 30 million has been funded, which will take us to the IND stage for this. And we are super excited about this program, and we started to have, before that, I want to acknowledge two of our colleagues, two of our partners in this program, otherwise it won't be fair. So we have UC San Diego, Professor Shao Chen, who is a bioengineering pioneer, who has spent a number of years trying to develop this kind of system. Now we combine the effort with the Chen Lab and a company called CellLinks, which is now under a bigger group, who has perfected a DLP photo light-enabled for printing of human tissues. So we try to bring all these assets together, try to make this program work. And we are, as I told you, super excited. In this slide is a very early data that this kind of co-culture system by which we can put blood vessels and things, and how cells can be more functionally mature and more physiologically relevant. I won't go into detail because it's not the focus of a talk. So in the rest few minutes of my talk, I'm going to spend on the beta cell program. And this, as I told you, we don't do like many others like SEMA or Vertex or other companies do. We try to do by mRNA. Everything is driven by mRNA. And we try to produce spheroids rather than single monolayer cultures. So what distinguishes us is, as I mentioned in this slide, is we produce a fairly homogeneous population of cells which are double positive for insulin and C-peptide. And we are at a stage we can produce about 2 billion cells, which is around the dose which are planning in patients. And our product has, product has shown consistently in vitro GSIS, glucose-stimulated insulin secretion so far. I'm going to show you some data regarding that. And the spheroid formation has certain advantages, not only in the functionality, but also in the transplantation outcome and how physiologically relevant these things can work inside the organisms. So how does the differentiation program look like? We try to expand the iPSCs from about 2 million cells to about 300 million cells, and then drive with a specific mRNA cocktail into definitive endoderm, pancreatic progenitors, endodermal progenitors, and then finally towards the beta cell at a scale of about 2.5 billion at the end, where we hope to dose the patients with about 0.5 billion each. Yeah, as I told you, we drive things with mRNA. And this is a 16-day protocol by which we sequentially introduce different transcription factors into the cell to drive specifically to, say, first definitive endoderm, then go into the pancreatic endoderm, and then subsequently into other cell types. And this is a fairly worked out protocol, and we are very confident that we are ready to go into the IND enabling stage, which we have started already. So as I told you, a proof of our differentiation programs, initially the cells started expressing the specific markers like FOXA2 and CXCR4, which is the endodermal, definitive endodermal marker, and they transition into a stage where they form like these islet-like clusters in the cell, and which we allow them to float into spheroids, which is again similar dimensions to the islet cells, human islet cells, as shown in this picture. We select for this size because of certain reasons, specifically because of their physiological competence at this size. And I can't go in details right now, but I'll be glad to explain why it is. As the cells transition through this, they express key markers, early markers like, mid markers like NKX6.1, which is a critical marker, I would say, in this beta cell differentiation program, because timing of NKX6.1 is very critical. If committed pancreatic progenitors express NKX6.1, then those cells are likely to have beta cells producing specifically insulin rather than polyhormonal cells. But if they continue to express, then you end up with beta cells which is with no functionality. And this is in our experience and other experience, it is being documented. So because of our mRNA technology, we are able to tightly control this expression. And that's one reason we get a fairly homogeneous population of beta cells. On the right panel is insulin-producing sections through the spheroids. And the cells are, what is more important is that not only insulin, but the key factors, Douglas Melton and others have published immensely on this regarding how cells producing insulin, C-peptide, NKX6.1, PDX1, co-expressing cells are so important when it comes to really mature beta cells. And that is what we see in this flow cytometry analysis. We started looking at the cells in the glucose response. We can repeatedly do it over a period and with a washout at the end with KCl, with depolarization. But what is shown is a single cycle where they can respond to glucose over a glucose challenge and produce insulin as expected of a primary cell. We can do it, we can culture the cells over a period of time, almost 3 months in the adherent format, and the cells keep responding in the similar manner as I explained. We started looking at early POC in animals with STC, which is a chemical-induced diabetic model. And as shown in this figure, over a 9-month period, we have consistent normoglycemia in the mice. This not only in the one mice, but in number of mice. The data shown is from one mice here. And that mice, we also, upon glucose challenges, it responds very nicely as expected, as shown in the right side of right graph. And after a 9-month period, when we section through them, their graphs survive very well. They have human antigens expressing as well as the insulin expressing in them. And as I told you, this is not only one mice, but multiple mice we have dosed, and they are consistent across the board. So not only we did with a chemical model, we also looked at NKG Akita model, which is a mutant mice having insulin mutation in it, which is a progressive diabetes. And in these mice, you don't need immunosuppression because they are already in the NKG background. So as you see, not only we have in the chemical model, we also see the same kind of response in these mouse, and we are very excited about it when we started moving into the IND-enabling activities. So as I told you, our goal is actually individualized therapy, autologous approach. Even though our approach is also amenable for allogenic, but our approach primarily is targeted for autologous. And we believe we can do that because of a number of reasons. Because we have a unique GMP capabilities, we have consistent production of A-beta spheroids, we have tested our methodology in a number of donors, including 10 donor lines in which we have tested and confirmed the consistency across producing the A-beta spheroids. We have about 50 donors recruited for this program. We can produce it at a dose which is generally acceptable in the field for a right dosage, about 250 million cells. And we also have started making progress towards cryopreservation, storage, shipping processes, everything validated for the future clinical trials. We are agnostic about the risk, and our risk mitigation plans start right from the collection of the cells into producing into final transplant. I won't go into details because we are going to run out of time. And finally, our initial goal is for type 3C diabetes, which is actually people undergoing pancreatectomy after pancreatic cancer and other and chronic pancreatitis. We have a partner in South Korea, SCM Biosciences, which incidentally we made the deal at this meeting, a number of years ago. They are going to do the clinical trials on the type 3C patient, but eventually we want to do in type 1 diabetes, for which we are collaborating with a company in Canada for producing the encapsulation device. We have started testing 1-year testing of that device in the mouse models. Is very encouraging, and we are very excited about this opportunity. Next meeting, I hope to give you more updates on how we are progressing in IND. Hopefully, we are planning to file an IND in 2 years from now. So for any questions or collaborations, please feel free to reach out at my email or at the general email at info@allelebiotech.com. Thank you very much.